Welcome back to the Sans Channel podcast, where we discuss the coolest scientific papers and preprints of our day.
Great to be back.
If you're finding these deep dives into the science as fascinating as we do, please take a moment to like this and subscribe to the channel. It really helps us bring you more of this content.
It really does. So today we're venturing into a realm that I know is of great interest to, well, pretty much everyone. The biology of aging.
Ah, yes, the quest for the fountain of youth, scientifically speaking.
Sort of. Specifically, we're diving into a study that explores the remarkable ability of the nematode C. elegans.
The little worm.
That's the one. Its ability to not just slow down aging, but potentially reverse it under certain conditions.
The elegans, yeah, the absolute workhorse of aging research. And you're right, the idea of, putting the brakes on aging or even turning back the clock on age-related decline, that captures everyone's imagination.
It's a universal quest, isn't it?
Totally.
And what makes C. elegans such a powerful model for this quest is its inherent flexibility and lifespan. Its lifespan plasticity.
Meaning you can really stretch how long they live.
Exactly. We can observe really dramatic extensions of life just by tweaking their genes or their environment. It's a goldmine for understanding the fundamental mechanisms that govern aging.
Okay, so this particular study focuses on something called dauer diapause. Sounds technical.
It is, but it's a fascinating biological trick they've evolved. Dauer diapause is a naturally occurring state, C, elegans enters when faced with stress.
Like what kind of stress?
Things like overcrowding or maybe food scarcity. Think of it as a survival mode, almost a form of suspended animation.
Okay.
During this dauer state, the worm's metabolism is significantly suppressed, and crucially for our discussion, its lifespan is dramatically extended.
So it's like they press pause on their development and aging, just to, weather the storm.
That's a good way to put it. They hit pause, and when conditions improve, they can just pick up where they left off.
And live a normal life afterwards.
That's the remarkable part. Upon exiting the dauer state, these worms resume their development, they become reproductively active, and live out a typical adult lifespan.
Even if they spent ages in that pause state.
Exactly. Despite having spent potentially an extended period in this arrested state, chronologically they might be quite old, but biologically, they seem to have been preserved somehow.
Wow. Okay, so this study dug into what's happening at the molecular level, gene expression you mentioned, and something called the bit age clock.
Yes, the Bitty age clock. What's particularly insightful here is how the researchers quantified this aging process.
How does that clock work?
It's a sophisticated tool, really. It looks at the overall pattern of gene activity, the transcriptome within the C. elegans cells.
So which genes are on or off, and how much?
Pretty much. This whole pattern serves as a molecular signature that correlates really well with the worm's biological age. By analyzing the levels of different RNA molecules, they can essentially estimate how old the worm is biologically.
Which, as you said with dauer, isn't always tied to its actual birthday, its chronological age.
Precisely.
So they used this bitten age clock on the dauer worms. Yeah. And did their biological age slow down?
It absolutely did. That was a key finding. Yeah. The biological age tracked by the Bitney age clock increased much, much more slowly in worms during the dauer stage compared to the passage of chronological time.
The aging clock was just ticking super slowly.
But effectively running at a snail's pace, yeah. But here's where it gets really interesting.
Okay.
Upon the worms emerging from dauer, the bit age clock indicated an actual reversal of age.
Wait, hold on. Reversal. As in going backwards? Their biological age actually went down. That sounds almost unbelievable.
It does sound dramatic, doesn't it? But that's precisely the core of what makes this study so compelling, and that's what we want to unpack in this deep dive.
Right. How is that even possible?
Well, the goal here is to understand the molecular mechanisms that C. elegans uses to achieve this slowing and this apparent reversal of aging during and after dauer. By examining the changes in which genes are active and, to what extent we can begin to piece together the biological processes behind this remarkable feat.
It's about figuring out how they hit the brakes and then somehow reset the clock without changing what the cells fundamentally are.
Exactly that, all without fundamentally altering their cellular identity. Understanding this in a relatively simple organism like C. elegans could offer some truly profound insights.
Insights for us, for human aging.
Potentially, yes. Insights into healthy aging in more complex organisms, maybe including us.
So we're not just talking about living longer. but living healthier for longer.
Yeah.
The health span idea.
That's the real prize, isn't it?
It really is.
And the dauer state in C. elegans presents us with this naturally evolved example of how an organism can achieve that state of extended youthfulness.
Okay, so let's get into the specifics. What did the study actually find? What molecular changes happened during dauer that might explain the slowed aging?
Right, so the analysis of the transcriptome, the gene activity during dauer, showed pretty significant shifts in metabolism.
Which makes sense if they're suspended, right? Less energy needed.
Exactly. Not surprisingly, many metabolic pathways were downregulated. This reduction in energy expenditure fits with the broader idea in aging research that a reduced metabolic rate can often correlate with increased longevity.
Like calorie restriction effects we see in other animals. Less metabolic activity could mean less cellular wear and tear over time.
That's definitely a key part of the picture, yeah. However, what's particularly noteworthy is that while overall metabolic activity was reduced, genes involved in crucial cellular maintenance processes were actually upregulated.
Maintenance. What kind of maintenance?
Specifically, protein quality control mechanisms. Things like the proteasome and autophagy.
The cellular recycling and waste disposal systems?
Yeah.
Getting rid of junk proteins and damaged bits.
Precisely. This upregulation aligns really well with the broader understanding of autophagy as a key longevity pathway across many species. It helps clear out the damaged cellular components that tend to accumulate with age.
So even though they're in this low energy state, they're actively investing in cleanup.
It suggests exactly that. They're prioritizing repair and maintenance, likely to ensure they can successfully resume development in good condition when the environment gets better again.
Okay, that makes a lot of sense. A period of dormancy that's also like a deep clean.
Right.
What about when they exit dauer? When the supposed age reversal happens, what changes then?
Right, the exit. Upon exiting dauer, the researchers observed, well, first, a general reactivation of metabolic pathways.
Makes sense. They need energy to grow again?
Logical, yeah. They need to ramp up energy production. But interestingly, the activity of the proteasome, the protein quality control system we just mentioned, showed a particularly dynamic pattern during this exit phase.
Dynamic how? It was upregulated during dauer, so what happens when they come out? Does it stay high?
Well, it depended. The pattern of proteasome gene expression upon exit seemed to depend on how long the worm had been in the dauer state.
Oh, really?
Yeah. In worms that had a relatively short dauer period, maybe just a few days, proteasomal genes were actually upregulated again when they exited.
So an extra boost for cleanup on the way out.
Suggests maybe a need to further enhance protein quality control for recovery, right? However, in worms that had been in dauer for a longer time, weeks perhaps, where proteasome activity was already high, these genes were either not significantly upregulated further or in some cases even downregulated upon exit.
So the length of the dormancy changes the recovery strategy.
It appears so. It seems the duration influences the specific molecular recovery strategies they employ.
That's a fascinating nuance. It's not just one switch, it's tuned based on their recent history.
Exactly. It really underscores the intricate nature of this rejuvenation process. The study also looked into something else quite specific, a phenomenon known as gene length-dependent transcription decline, or GTLD.
GTLD, okay, what's that?
Think of it like this. In normally aging cells, across different species actually, longer genes tend to get expressed at lower levels as the organism gets older.
Why?
It's thought to be related to the accumulation of damage along the DNA strand, like little roadblocks. These can impede the RNA polymerase, the machinery that reads the gene, especially when it has to travel a long distance down a long gene.
I see. So it's harder to make the full copy of a long gene if there's damage scattered along it?
That's the idea. And did the dauer state affect this? Were they somehow shielded from this age-related transcription decline?
Good question. Were they?
Well, we know dauer larvae are generally more resistant to various stresses, including DNA damage from things like UV radiation.
Yeah.
And the researchers found that even when they zapped dauer worms with UV light, which causes these transcription-blocking lesions, the worms were highly efficient at repairing this damage, especially during the exit phase from dauer.
So they're not just tough against the initial hit, but they actively fix the consequences afterwards, particularly when waking up.
Precisely. The study demonstrated that during the exit from dauer, the worms effectively restored the integrity of their transcription process. They resolved that gene length-dependent transcription decline caused by the UV damage.
Wow.
It strongly suggests that this exit process triggers really robust DNA repair mechanisms. It ensures their genetic blueprint is in good working order for resuming development and heading towards a healthy adulthood.
It's almost like the dauer state isn't just a pause, but a window for some profound cellular R&R repair and restoration.
That's a great way to put it, ensuring that when life resumes, it does so with a remarkably refreshed, and rejuvenated cellular machinery.
So this study paints a pretty compelling picture then. Dauer diapause isn't just slowed aging,
But potentially actual biological age reversal, yeah. Characterized by this careful management of metabolism, enhanced protein quality control, and these highly effective mechanisms for maintaining genomic stability, especially during that transition back to active life.
Okay, so let's boil it down. For our listener who's been following along, what are the key takeaways from this deep dive into the C. elegans dauer study?
Right, let's recap the core findings. First, C. elegans can enter this dauer state under stress, becoming exceptionally long-lived, but, crucially, retaining their reproductive capacity after exiting.
They don't sacrifice fertility for longevity in this case.
Correct. Second, their biological age, measured by that bitty age clock, significantly decelerates during dauer and, remarkably, appears to reverse upon their return to normal development.
The headline finding, really.
Definitely. Third, this rejuvenation doesn't seem to involve making brand new cells through division, but rather a restoration or repair of existing terminally differentiated cells.
So fixing what's already there.
Seems like it. Fourth, metabolism generally slows down during dauer, while those key protein quality control systems, the proteasome autophagy, are enhanced. Housekeeping gets a boost.
Got it.
Fifth, when they exit dauer, metabolism fires back up, and the proteasome activity shows that dynamic pattern we talked about, influenced by how long they were dormant.
The duration matters.
And finally, dauer larvae are resilient to DNA damage and can efficiently repair it, especially during the exit phase, which helps maintain the accuracy of their gene expression, resolving that GTLD issue.
It really does highlight the dauer state as this extraordinary natural model system for understanding longevity and rejuvenation.
Indeed. This research provides really valuable clues into how an organism can naturally achieve not just slowing, but even reversal of aging at a molecular level.
Which brings us to the big so what? The final provocative thought for our listener.
Right. Could understanding these intricate mechanisms of age reversal in a tiny worm like C. elegans ultimately pave the way for new strategies? Strategies to promote healthy aging and treat age-related diseases in humans.
And maybe offering us approaches that are, more natural or less drastic than some of the cellular reprogramming ideas out there.
Exactly. Could deciphering these secrets unlock novel strategies for extending human health span? Combating age-related decline? Perhaps using gentler, more physiologically natural approaches than we currently envision.
It's certainly a provocative thought to chew on. Absolutely fascinating stuff.
It really is. A lot to think about.
Well, that brings us to the conclusion of another, I think, deeply insightful dive.
Agreed. Always a pleasure.
Please don't forget to like the video and subscribe if you want more content like this.